Hello, I'm Kim Fan and I am here with Neha Malassari, Elijah Roberts, and Lauren Crowhurst to present our findings on found phages, dynamite, and casserole. SEA-PHAGES, or Science Education Alliance, Phage Hunters Advancing Genomics and Evolutionary Science, is dedicated to expanding the scientific community's knowledge of bacteriophages, which are viruses capable of infecting bacteria. The Arthobacter phage strain contains diverse genomes with mechanisms for phage replication and lysogeny. Arthobacter is a resilient strain of bacteria primarily found in soil. It can survive long periods of starvation, temperature shifts, ionizing radiation, oxygen radicals, and toxic chemicals. The contribution to phage knowledge through SEA-PHAGES program is an integral part to many sectors of research and has previously resulted in multiple biotechnology advances such as phage therapy and CRISPR-Cas9. The Baylor cohort of SEA-PHAGES used the host Arthobacter to isolate and cultivate Arthobacter phages, dynamite, and casserole. These phages were isolated and collected from the Baylor University campus. Phage information was submitted to phagesdb.org, and bioinformatics tools were used to annotate each genome to determine the possible gene functions.
I'm Neha, and our study consisted of two sections, a wet lab and a dry lab. The wet lab portion of the study consisted of isolation, purification, amplification, and DNA extraction procedures. Soil samples were collected and purified using soil washing, cereal dilutions, spot tests, and plaque assays. The plaques were picked to create web plates, which were then flooded with buffer to create a high-titer lysate. Then the promega DNA binding resin was used to extract the phage DNA. Characterization was then conducted using tape measure gene PCR cluster analysis and gel electrophoresis. Finally, transmission electron microscopy was used to determine phage morphology. The dry lab portion of our study focused on annotating phage genomes and predicting gene locations and putative functions of the phages using Glimmer, GeneMark, NCBI, PhagesDB, HHPred, Starterator, and Famerator.
Hello, I'm Elijah, and as mentioned previously, during this study we successfully isolated two Arthobacter phages, named Dynamite and Casserole. Dynamite, which is pictured above, is a lytic Cyphoviridae phage with a long contractile tail. It forms plaques that are about 0.9 millimeters in diameter, and we determined it to be in the AM cluster as per our PCR cluster analysis, which we later confirmed when the phage was sequenced. Dynamite's genome is 47,846 base pairs long, and these make up a total of 100 protein-encoding genes. 99 of those genes are transcribed in the forward direction, and 1 is transcribed in the reverse direction. Its genome has a GC content of 45.3%, and it has a 3' sticky overhang at the ends. Our second phage, shown below, we named Casserole. Casserole is a lytic Podoviridae phage with a short, non-contractile tail. It forms plaques of two distinct sizes, one a bit more than 1 millimeter in diameter and one a bit less than 1 millimeter in diameter, and we identified it as being in the AV cluster. Its genome length is 46,556 base pairs, and we identified 58 genes, 30 forward and 28 reverse. We've called a function for 19 of these genes, and we've determined 8 of them to be orphams, with no known analogous genes. Casserole's GC content is 45.8%, and its genome ends in a direct terminal repeat.
Hi, I'm Lauren. In conclusion, our experiments fulfilled the purpose of the SEA-PHAGES program in that we isolated, purified, amplified, characterized, and annotated phages, dynamite, and casserole. Both of the phages are lytic for the host Arthobacter, but they have many differences both genetically and morphologically. Dynamite is the more typical of the two phages as it is a Cyphoviridae in the AM cluster. What is interesting about dynamite is that it shares all but 2 base pairs of its genome with phage Napoleon B. In contrast, phage casserole is very unique. It is a Podoviridae member of the AV cluster, and its genome contains an unordinary number of reverse genes, orphams, and bacterial genes that seemingly belong to a host other than Arthobacter. Both casserole and dynamite have interesting characteristics that deserve further exploration. For example, genes 18 and 19 in phage casserole match the bacteria R. pyridinivorans. In the future, casserole may be examined in wet lab to see if it is compatible with other hosts or if the genes like 18 and 19 were picked up from the environment rather than an alternate host. Once the gene annotations performed by this study are checked and confirmed, they will be published for anyone to access. All of the information regarding phage dynamite and phage casserole can be found on the actinobacteriophage database, which is accessible to anyone who is interested in learning more about bacteriophages. Dynamite and casserole may be used in further studies regarding Arthobacter. We would like to thank the HHMI SEA-PHAGES, Baylor University, and Dr. Tamara Adair.